Science? All right, yeah, excellent, excellent. Well, good evening and welcome to the South Carolina Aquarium for Season 7, Episode 1 of the Holland Lifelong Learning season. Season 7, do you believe that? I mean, we're right up there with any good Netflix docu-series right now, so thanks for coming out. I see a lot of great to have your support, some new faces, so appreciate your support. All right, we're excited to kick off the year in hybrid fashion tonight. So looking to have some fun and fulfill our vision of connecting people to water, wildlife, and wild places. In addition to our little coffee shop audience that we have here in the lower Great Ocean tank, we had about 100 registrants over there tuning in virtually. So hello, wherever you are. As an organization, we are grateful for your continued support. So thank you with that. This series has become a signature program for the South Carolina Aquarium. And really it was meant and designed to connect our community with scientists and the science that's right in our backyard. And that's what we're here to highlight tonight. We do want to give a big thank you for the generous support of Mary and Mason Holland, as well as Chris Cocker over at Ameriprise Financial Services. Tonight's chat, here we go. Tonight's chat is literally a no-brainer. In fact, not only is tonight's talk a no-brainer, it's also heartless, bloodless, and boneless. Tonight, our speakers will focus on an animal that is made up of about 95% water and is essentially a floating mouth out there in our oceans, the jelly. Tonight, we get to feature Dr. Lara Treible, a postdoctorate research associate at University of Georgia Skidaway Institute of Oceanography in lovely Savannah, Georgia. She received her bachelor's degree in environmental science from the University of Delaware, her master's in marine science from Stony Brook University, and PhD from the University of North Carolina, Wilmington. We always like to share that stuff because we have young students, I see you back there, and we have young students joining in virtually that might be thinking about science careers. So it's great to share the road, the path that y'all have taken as scientists. You guys are the future. You gals are the future. So always good to know. All right. Dr. Treible's research focuses on gelatinous zooplankton, jellies. Research is looking at population dynamics, changes in communities, and things that drive those changes. Her research also focuses on the early life stages of jellies and the response to various environmental conditions, such as temperature, low oxygen levels, and coastal acidification. Shannon Howard is one of our own, a senior biologist too, here at the South Carolina Aquarium. She moved to Charleston to pursue a career in conservation after receiving her Bachelor of Science degree in zoology from, I think you have to say, the Ohio State University. Is that how you do that? All right. She quickly began volunteering for the Folly Beach Sea Turtle Watch program in the United States Fish and Wildlife Service, where she continues to participate in sea turtle conservation to this day. Early mornings, right? In 2003, she began volunteering for the South Carolina Aquarium and was hired as a part-time aquarist and then full-time in 2006. During her career at the aquarium, she has worked with sea turtles, octopuses, seahorses, jellies, and many other marine animals. Shannon is an advanced open water rescue diver and licensed motorboat operator, assisting with our dive team and animal collections team here at South Carolina Aquarium. So we're here to talk about science. We're here to chill, to have some fun, sit back, enjoy our casual conversation with these two wonderful ambassadors of science. Without further ado, our very own Shannon Howard.
All right. Can y'all hear me? Oh. Mic drop. All right, so no pun intended. Are y'all ready to talk some smack? So a smack is a group of jellies. So this is a Pacific sea nettle. So you can see that's a smack, a Pacific sea nettle. So jellies are gelatinous zooplankton. So all jellies are gelatinous zooplankton, but not all gelatinous zooplankton are jellies. So these animals are, as Brian said, they're very simple, very delicate animals. They have more transparent bodies. They're 95% water, give or take. They live in the open ocean. So all zooplankton is, they're ocean drifters. You can think of them as that. They're not very strong swimmers. And this is a little plug. In Laura's talk, you'll see some more photos similar to that. But the diversity here is immense. There's over 2,000 species. Thanks to deep ocean exploration, they are IDing new species every day. It's really is amazing. And the next photos, I'm just going to show you examples of the animals. Obviously not all of them, but I want to show you the different body shapes, sizes, colors. These are so diverse that they're the most common animal in the ocean, hands down. The most common animal, they live in every single ocean. There's some that live in freshwater even, and they are at every depth. So from the surface down to the depth of the ocean. So first we're going to talk about the tunicates. So most people think of tunicates as sea squirts. They are our relatives. So they are in the same phylum as we are. We're in the phylum Chordata, and so are they. And there are pelagic tunicates. So the first group I'm going to focus on are the Cytos. These are considered the ocean's vacuum cleaners. All of these guys have barrel-shaped bodies. Well, this group anyway, has barrel-shaped bodies, and they are filtering food from the water at an enormous rate. So they can have really long chains. So the chains that you see are multiple animals per chain, and they can also be in singles as well. And those chains can be as long as a football field. It's really is amazing. So Deliolids are next. Laura's going to go into more detail about the Deliolids, but again, you can see the barrel shape and their filter feeders as well. The top right photo is actually a reproductive Deliolid. And then the pyrostomes are next. So these are considered sea pickles, and they are bioluminescent. So their colors are really amazing. And if you look in the photos, every little bump that you see is an animal. So they're colonial animals. And I love this picture. So that's a leatherback sea turtle. Leatherbacks can get up to 8 feet long, and that's a pyrostome that it's eating. So these jellies really have a huge role. They are food sources for so many animals. And it's just amazing. To me, it's amazing. So the larvaceans are next. And these are really small animals. They kind of look like a larva or a tadpole. And one thing, so they're also tunicates. They're in a different class than the ones I showed you previously. But in the right photo, the small animal is in the center, and that big thing that kind of looks like lungs around it, that's a mucus sac that they produce to filter feed. So next are the ctenophores. These are the comb jellies. So they get their name because they have rows of cilia that help them move through the water. They can be tiny. They can also be larger. They're some of the largest animals that move with cilia. And the cilia can refract light. So when you look at a comb jelly, usually you'll see a rainbow effect. That's the cilia moving, refracting that light. Some of them, not all of them, are bioluminescent. So the first class is the Temeculata. Again, these are in all oceans at all depths. New species are being discovered regularly. The bottom center is a newer species. So the Temeculata, what it kind of tells you is they have tentacles. So they have long tentacles that can retract. And those tentacles don't have venom. They have sticky cells called coloblasts that they use to collect their prey with. And then again, more of these species. And where you see the red animals, those are more deep sea animals, because red light at depth, that animal it disappears. So it gives those animals camouflage in the depths of the ocean. And this is another one I wanted to show you. It's called a Venus girdle, very different animal, and it does have cilia. It is a true comb jelly. And just to say, bioluminescence, you can only see it at night, and you can only see it when the animal is disturbed. So Nuda is the next class of the ctenophores. These do not have tentacles. They actually have a mouth. Very interesting. So the bottom left, you can see that mouth, and then the top center is a ctenophore that had eaten another jelly. So they eat other jellies, typically other ctenophores. And the colors on them are just amazing. So next up are the cnidarians. These are the stinging jellies. And the ones I'm gonna focus on are the free swimming organisms, because they're the zooplankton we're focusing on. They're the true jellies. So hydrozoans are the first class I want to talk about. And the one on the left has been recently identified. And the one in the bottom center, that guy has a fish in its tentacles. And here are more hydrozoans. The one on the left is a crystal jelly. We do have these locally. These actually eat other jellies as well, similar to the Nuda class of the ctenophores. The bottom center is a flower hat jelly, very beautiful. And the right is a freshwater, one of the few freshwater species of jellies that exist. And the top center may look familiar to you. That's the immortal jellyfish. So that animal under stressors, if it gets old, it can actually regress to the polyp phase. And that polyp phase will produce more medusa that are identical to the original animal. So more hydrozoans, these guys are colonial hydrozoans. So like the pyrostome, you have so many tiny little organisms making up the whole individual. And then you have different groups of those organisms that have different functions in the body. So we have the blue button on the left, and on the top, I'm sure everyone knows what that is. That is a Portuguese manophore. And I will just say, they are not always deadly. Several people have gotten stung by them and they're fine. I'm going to talk about this thing a little bit later. In the bottom center is a deeper ocean one that you can see it's using jet propulsion. So the cubos, the box jellies, these are probably the ones with the most venom. So you have the irukandji on the left and flickeri on the right. Those two species are pretty dangerous. Most of them are in Australia, so we don't have to worry about that as much. The top center is a native species to us. And the bottom I wanted to show you, so the irukandji can fit in a tiny little vial. In that bottom one, you can see the 5 millimeter, that is a pretty small animal. So these can be very small. The flickeri on the right, those can actually be about the size of an average human's head. So the cyphozoans, so these are the true jellies. These are the jellies that have the bell, or that umbrella-shaped bell. And these are the crown jellies. So they get their name because they have a groove around the top of their bell, so hence the name. And the bottom animals are more deep sea jellies, and the top ones are thimble jellies, so they can be higher up in the water column. And another group is cyphozoans. These are actually the edible group of cyphozoans. If you've heard of jellyfish noodles, they get them primarily from these animals. The cannonball jelly is on the bottom right, and that is a local species to us. Please, if you're out on the beach, don't throw them at each other. But these are just amazing. So you see how different they are. These don't have true tentacles. They more have oral arms. modified oral arms that you can see that look kind of like a flower. The bottom left is a Cassiopeia. So talking about the roles that these jellies play, those animals have symbiotic algae living on them, mainly in their tentacles, and they use that to feed themselves. So very interesting. So next are the Cyphozoans, again, Cyphozoans, continuing, but these are the animals that everyone really truly recognizes as a jelly. We have a Pacific moon jelly on the top left, an egg yolk jelly bottom left. Those are really pretty. Drymonema larseni, so the one in the center on the bottom, that one was reclassified in, I believe it was 2014, and we collected one when we were on a collecting trip. We collected one by accident. It was attached to our moon jellies, and I didn't know it. So I left for the weekend, came in on Monday. I had this big kind of, I guess, softball-sized jelly. I didn't know what it was. I went to my coworkers and I was saying, hey, who put this jellyfish in my tank? I don't know what this is. And I found out it was a pink meanie. It was this Drymonema larseni. And I was able to send it to a researcher who was doing genetics research on this animal. So he was able to recently reclassify it with genetics, which is pretty cool. That's one of my favorite stories. So the common species we display, we have two exhibits. So the moon jelly exhibit on the left, and then the one on the right, we rotate species in. The sea walnut is a ctenophore, a cone jelly we display on occasion. These are in our waters in the spring and in the fall, usually. The lion's mane jelly, they're beautiful. Like they're one of my favorite jellies, hands down. And if you look at this picture, look at all of these little fish. So people don't realize a lot of larval fish will use jellies that are venomous as protection. Not all venomous jellies, but they will just use them as protection from their predators. So jellies don't eat every fish that they come in contact with, unless that fish isn't smart and hits a tentacle. So this animal actually, they don't get big in captivity and they don't like warmer water as much. So I've never seen them locally bigger than the size of a 5-gallon bucket. But in the colder waters, they can get, I think the largest one on record was 120 feet long. That's longer than a blue whale. It really is amazing. And then right now we have Atlantic sea nettles displayed. And of course, our moon jelly. So this is the species that we display year round and we culture here at the aquarium. And these are some local ones offshore to us. This is my photo. So what do we house these animals in? We need specialized tanks. These tanks are called Plankton Chryslers. So if you look, it's circular, so it gives nice, even flow. There's no corners for them to rub up against. There's nothing in with them for them to rub up against, and they can't get stuck to any intakes. So anatomy. Again, this is a moon jelly. So they have radial symmetry. So you can cut them into 4 pieces and they will be equal. They have a nerve net. So like Brian was saying, they don't have a heart. They don't have any major organs, very simple animal. So they have a nerve net as their main sensory structure. And they have oral arms. So you can see 4 kind of ribbon-like structures. Those are oral arms for feeding, obviously. They have gonads. So there's that 4-leaf clover, that's where their gonads are. You can have male and female medusa. I think a lot of people don't realize that. The mouth is right in the center at the base of the oral arm. So the oral arms the tentacles will collect that food. It will transfer to the oral arm, and that will move it up to the mouth and the stomach. The rhopalium, those are sensory organs. They have 8 of them. In between each lobe is one of those sensory organs, so they use that along with the nerve net. And the radial canal and the circular canals that they have just help with nutrient transport. And then their tentacles are right along the margin. So again, this is moon jelly anatomy. It's not necessarily the same for the other jellies. So let's talk Pneumaticists. Let's talk tentacles. So, let me see. So this is a syndocyte. This structure right here is a syndocyte. That's a cell. Imagine a tentacle running right along this line, and embedded in that cell is a nematocyst, and there's a coiled filament in there that's barred, and it contains the venom. And here's a trigger that's called a syndocell. So if something triggers that, it'll cause the nematocyst to fire. And here is a tentacle. I love this photo. And you can see the syndocyte right there and the trigger right there. And then this is a photo of a tentacle with the nematocyst fired. So a lot of little kids get really upset when they get stung by a jelly. They think it was on purpose. These animals don't have brains. If something hits that tentacle, it'll cause that nematocyst to fire. It's their only way to defend themselves. Again, they're very, very delicate. And it's also the way they feed. So the life cycle. These are baby moon jellies called Ophyra. Love that photo. And this is the life cycle. So you have the male and female adult medusa. When the egg is fertilized, they will turn into what we call planula larvae, and they will disperse off of the oral arms and then turn to a polyp. I have time. I'm gonna start speeding up. I'm sorry. So then they'll turn into a strobila. So under certain conditions, it'll cause them to start this process called strobulation, and that will turn into the Ophyra, which will grow up to be the male and female adult medusa. So strobulation is an asexual reproductive stage where a polyp turns into a medusa. And here you can see the stages. And then this photo is just to show you that the polyps can also reproduce asexually. So there are budding polyps. If you look closely, you can see those budding polyps there. And then the strobila right here. So our moon culture, this is our current moon culture tray. So the polyp bins are about the size of a loaf of bread. You've got the catch bin, and then you have the grow out bins where they grow up. And then once they're about a dime to a nickel size, they will move on to a chrysal, like I showed you before. So these are about a day old Ophyra, give or take a few days. So the way we do that is we alter the temperature. So we will lower the temperature. We keep them at 70 degrees. We'll lower it about 5 to 10 degrees for a week, bring it back up, and within 2 weeks, we usually have Ophyra. They don't like to go off more than a year in general. But I can also change the salinity, so the level of the salt in the water, and I can also add iodide to try to stress them to get them producing. So blooms. Blooms is always a hot topic with jellies. So blooms are a natural event in these animals' life cycle. They happen on a cyclical 20-year cycle generally. Are blooms on the rise? There needs to be more research. We are not certain yet, but we do know that the oceans are under threat, and these threats affect the jellies. So marine debris, wastewater runoff, and overfishing. Marine debris, hard-sided plastics, those polyps can live on. So you have a lot of surface area for the polyps to grow. And then you have wastewater runoff, forms of pollution, sewer, fertilizer runoff. Those can produce nutrients that will stress these jellies, that will cause them to start strobulating. And then you have overfishing. So if you have a lot of the competition that the jellies have for food are taken out of the equation, it could cause them to grow exponentially. So you kind of see the theme here. So I see these guys as biological indicators. Every bloom that we think is out of balance could be a signal, hey, something's going on here. What can we do to change this? It's up to us, because all of those ocean threats, those are usually caused by humans, right? So in essence, I would like you guys to change. If you have a negative view of jellies, which there's a lot of negativity out there, I would like you to change your view and see them as I do, which are ambassadors for healthy oceans. Thank you. Thank you so much, Shannon, for getting us started off describing the different types of jellies and kind of ending on this point of seeing jellies as ambassadors for healthy oceans. So just to reintroduce myself, I'm Dr. Lara Treible. I'm a postdoc at Skidaway. And today I'm going to be talking about some of my research on gelatinous zooplankton. And so a great transition from what Shannon was just talking about is that most people, when they think of gelatinous zooplankton or they think of jellyfish, they think of the negatives, right? Most people think about getting stung. How many people have been stung by a jellyfish? Yeah, a lot of people. So people tend to think of, what I'm showing here, potentially getting stung, maybe jellyfish washing up on the beach. This image is actually from Hilton Head, so it's relatively close to here. Maybe you guys have seen beaches that look like this before. But jellies are actually a really important part of ecosystems. Shannon has shown some pictures too of little fish living underneath the bells of jellies. They're a big part of the diet of some sea turtles, and humans can eat them as well. If you've ever been to an Asian restaurant that serves jellyfish salad, yes, that is actually made of jellyfish. So they're really important for humans and for healthy ecosystems. And so much of my research has been looking into the environment that leads to these jellyfish blooms or potentially things that might be negative impacts on these jellyfish. And so I'm really answering three major questions here: who, where, and why. And the who is what groups are there, what taxa. Shannon gave a really good overview of a bunch of different types of jellies, so I'm looking to see who's there. Where they are, so not just the general location, but also more specific locations like depths. And why are they there? The different environmental conditions that are leading to these distributions. And so through this, I use multiple approaches. Most of my dissertation work was experimental, so looking at potential environmental impacts, things usually associated with climate change. And my current postdoctoral work is more observational, so looking at the location, the abundance, figuring out what different environmental conditions are correlated with where these jellies are. And so I do just want to bring back up this scyphozoan life cycle. Shannon just went through that really well. But for my experimental work, I focused on these early life stages, so the polyps and the ephyra. And so here on the right, you can see a polyp that has produced some other small polyps through asexual budding. And then on the left here, you can see that process of strobilation. So those polyps are undergoing this process and producing ephyra, which are the baby jellies. And so we don't have a ton of time today, so I'm just going to talk about one experimental case study, and that is that of temperature. This figure that I'm showing here is the global temperature change from 1901 through 2020. And so as you can see here, most of the oceans are in red. That is an increase in temperature. And this is driven by increased atmospheric CO2. So we're putting more CO2, carbon dioxide, into the atmospheres. It's warming up our atmospheres, warming up our oceans. And so the question here is, will this lead to more jellyfish? There is kind of this thought that with warmer temperatures, we'd see more reproduction and thus potentially more jellyfish. But I wanted to dive into this and look at the effects of temperature on the reproduction of these early life stages. So focusing on the polyps and the production of the ephyra. And so for this experiment, I used 3 different species. I used the Aurelia, which is our moon jellyfish, and then 2 different species of sea nettle, the Atlantic and the Pacific sea nettle, and they were exposed to 4 different temperatures. And the reason we chose these 3 species is because they have different natural distributions and they're naturally exposed to different temperatures. Our moon jellies are pretty ubiquitous throughout our coastal oceans. So they've got a really wide range of different temperatures that they're naturally exposed to. The Atlantic nettles are found in the temperate to subtropical temperatures along the east coast of the US, and then our Pacific nettles are in a little bit colder, deeper water on the west coast. So they have these different natural temperature ranges. And so what I did was expose polyps of these different species to these different temperatures, and I collected 4 different types of data. I looked at budding of new polyps. So these polyps are asexually making new clones of themselves. I looked at how many were strobulating and how many ephyra they're producing. So how many are undergoing that process of producing baby jellyfish and how many babies they're producing, and the stage and timing. So how long does it take them to undergo that process? Now I'm going to show some of the results of this. They are going to be graphs, but I'm going to kind of walk you guys through what exactly these results mean. So the first is the budding or total polyps. So all of these graphs that I'm going to show along the bottom here, they start with cold on the left to warm on the right. And what we can see here is that our general trend is positive. Cold has less polyps, warm has more polyps. This is pretty much expected because an increase in temperature usually corresponds to an increase in metabolism. So we would maybe see an increase in their reproduction, pretty expected across all 3 of these species. Next is strobilation. So again, we have cold to the far left and warm to the far right. And here we see that the 5 degrees, or the coldest, was too cold for them to strobilate. Same thing with the warmest. 20 degrees was too warm for them. But these intermediate temperatures here, 10 and 15, we did see strobilation. And the interesting thing here is within these intermediate temperatures, it was the slightly cooler of the temperatures, so 10 degrees Celsius, where we actually saw all 3 species strobilate. So we had the most strobilation at that slightly cooler of the intermediate temperatures. And we saw something similar with how many ephyra were produced. Again, coldest was too cold, our warmest was too warm, and we got ephyra production in those intermediate temperatures, but we got the most at the slightly cooler of the temperatures. So that 10 degrees had the most ephyra produced. So we're seeing a little bit of a trend here. We see something similar with strobilation timing. So I've got kind of over time on the left in graphs, and then I've got it on the right in a table. But essentially the take home here is that at 10 degrees, all 3 species, it took about 3 weeks for them to start strobilating. All 3 species were strobilating about the same time. At 15 degrees, so a little bit warmer, we saw a delay in strobilation. So about 1 to 2 weeks later, those slightly warmer ones started to strobilate. So what does this all mean? Well, we saw a pretty solid trend across those results, that we need slight cooling for that process of strobilation, right? We needed it to not be at that warmest temperature. We needed that slight drop to that 10 degrees Celsius. And so when we had that slightly cooler temperature, we got earlier strobilation, and more ephyra. But the warming was good for more polyps or more budding. So potentially, if we're seeing temperatures continue on this trend, we might see a lot more polyps, but maybe not more of the adult populations that we might expect to see. Now, all of that is well and good. Experiments tell us a lot about these kind of potential scenarios, but it doesn't tell us exactly what is going on out in the field. What are these jellies doing right now? Where are they? And so to find where they are in the field, we have to go jellyfishing. I think when you mention jellyfishing, a lot of people automatically think SpongeBob. So we've got SpongeBob and Patrick here kind of going around with their nets collecting jellies. And it's not completely off of how we collect jellies. Typically, plankton in general are collected with plankton nets, as the pictures up here. But unfortunately for jellies, we can collect them in plankton nets. But they're very fragile. So here's a picture here on the right. This is a little ctenophore that was caught in a plankton net off the coast of Long Island. But they're very, very fragile. So you can imagine filtering a lot of water through a net is going to be a little bit damaging for especially these very, very fragile ones like these ctenophores.
Jellies can also be very patchy in their distributions, so nets are going to sample a large amount of water at once as you drag that net through the water, and so we're not really sure if we get a jelly in a net where it exactly came from, where exactly at that point in the water column did it come from. And so some of the work that I do now is working with optical or imaging systems. And imaging systems are really great because you can get that fine scale distribution. It's not just a net that jelly might have been anywhere in the water. We can know exactly where it is in the water column. Imaging systems are also not biased to certain groups. So jellies, a little bit fragile, not great in a net. Imaging systems are really excellent for sampling jellies. So the one that I'm showing you up here in the diagrams is called the ISIS, or the In Situ Ichthyoplankton Imaging System. Just a fancy name. It was created originally for sampling larval fish. That's that ichthyoplankton. It collects a lot of data. So it's about 16 to 17 images a second. That gives you about 2 terabytes of data every 3.5 hours. So we're collecting a ton of image data, and it's collecting environmental data. So things like temperature, salinity, chlorophyll, which is going to tell us how much algae is in the water, and oxygen. And so the ISIS, the picture here on the left is what the ISIS looks like. And then the diagram sort of on the center right is a schematic of how it actually images in the water. And it images through this process called shadow graph imagery. So you have this LED light in this top panel, and you have your camera there in the bottom. And essentially what it does is it uses mirrors to bounce that light through the water, and then it's captured by the camera. So it's taking a picture of the shadow of organisms. There are a lot of benefits to imaging in this way, and one of them is that it's really great for things that are transparent. So here we have a small flask. It has a small eel larvae at the bottom, not a gelatinous zooplankton, but it is transparent. And you can see it there circled, and it's really difficult to see because it's transparent, just really hard to see with the naked eye. But with a shadow graph system, that's what the exact same eel larvae looks like. So you can see how difficult this would be to see with a regular image and how great it looks in that shadow graph imagery. So to process these images, we go through a lot of steps. The raw image files look like this. It kind of looks like a lot of noise. You see those vertical lines through the image. We take it through a flat fielding procedure, which removes those vertical lines and makes it more black and white. And so that's what these look like. Then we send it through a computer program that segments. So it goes through and it picks out anything that it thinks could be an organism or a particle. And so the segmentation will actually cut out regions of interest or potential organisms. And then I go through them and identify them. So we end up with images that look like these. These are some examples of some of the jellies. And I sort through them to say, is this a jelly or not? And if it is a jelly, what type of jelly is it? So I'm going to show you some examples of some of the questions that we can answer with this imagery data. And all of these examples at the beginning are from the Gulf of Mexico, sampled with that ISIS. And so here you can see Shannon also showed some really cool pictures from the ISIS, actually from the Gulf of Mexico, just the different diversity of plankton that we get in this area. And so research questions, there's a ton of questions that we can answer, but some of the things that I'm interested in, one is habitat preference. And so in this top panel here, these are distributions of ctenophores. This bottom panel here is a hydromedusa, and every one of those little black dots is an individual. And you can see that they have very different habitat preferences. The ctenophores, especially in the center and right panel, are mostly configured to the top of the water column. We see them kind of aggregating near the surface. Whereas this Hydromedusa liriope, we see it kind of aggregates near the bottom in this location, near shore in this location, and we get very different distributions of these organisms. Other really cool research questions that we can answer is, we have exact locations in the water column, so we can see if they're migrating between daytime and nighttime. This again is a Hydromedusa called liriope, and that's one of our shadow graph pictures that you see there. And on the left-hand side is daytime and the right-hand side is nighttime. And that dashed blue line is the average depth. And so what we can see here pretty clearly is in the daytime, their average depth is much deeper in the water column than at nighttime when they're migrating up a little bit shallower. We also can see really cool interactions between organisms. And this isn't just for gelatinous plankton, this is for plankton in general that we're able to image. And we actually just recently published a paper, I think it came out just about a month ago, on interactions that we can see from these images. So that's what these figures are from. We can look at predator-prey interactions. And so you can see here we actually have some interactions of organisms consuming other organisms in the plankton. And we can see some instances of a previous predatory interaction. So there's actually a little bite taken out of this jelly right there that you can see in the images. We can also see commensalism or mutualism. So these are going to be interactions where both parties are going to benefit, or maybe just one party benefits, but the other one isn't harmed. And so we can see those along the bottom here. Again, examples of larval fishes associating with jellies. And so we've got our little fishes here that we see in our images. We can also see examples of life histories, so reproductive events or juveniles and adults. And so our top panel here, we can see juvenile moon jellies that are growing up. And then this bottom panel here is a reproductive event. So that's releasing a bunch of new individuals. So the current project that I'm working on is using this in situ imagery, working with Deliolids. So again, Shannon gave a really great intro to what Deliolids are. They're pelagic tunicates. I've got a little video here on the left that you can see two little Deliolids drifting around in a jar. But this project is looking at Deliolids and their kind of contribution to the microbial loop in ocean. So we call it Dole Microbe is the project. And so I'm based out of Savannah, or Skidaway more specifically. And so every month we go out sampling Deliolids for 2 days, sometimes a little bit longer. And this is where we go. We go here on our mid-shelf to the 25-meter and 45-meter isobaths. Essentially, those are just 25 meters and 45 meters deep. Here's our research team on one of our recent cruises. Now, when we go out sampling Deliolids, we don't have that big ISIS that I already showed you. We have a smaller imaging system. It's called the Mini Deep Focus Plankton Imager, MDPI. This is what it looks like. It has some really cool things on it. We have a CTD, which does salinity, temperature, and depth. It has a navigation box, so that's going to show you how flat it's flying, if it's nose up or nose down or side to side. It has a fluorometer that's determining chlorophyll or how much algae is in the water, and we have an oxygen sensor. And then in the front is our imaging. So we have a camera here on one side, we have our light source on the other, and then all of the plankton that are going in between those two tubes are going to be imaged. So this is how we deploy the MDPI. We send it off the back of our large research vessel, the RV Savannah, and we tow it in what we call a tow-yo fashion. So it's kind of yo-yoing up and down in the water column as it's being towed horizontally through the water. And this is what it looks like when we deploy the MDPI. We have our computers on board. You can see in the video on the left, it kind of just looks like you're like going through a snowstorm or maybe flying through space. But each one of those little particles is plankton. And that's why it's really helpful to have those programs that help us segment those out, because if we had to do all of that by hand, it would take forever. We do still use plankton nets. I did tell you guys earlier that they weren't great for sampling jellies, but we do still use them. One of the reasons that we still use them is because we want to collect things that we can look at when we're actually on the boat. When we deploy that imager off the back, we can't really see, like, hold or put into jars anything that's out there. We see it as it's going by. But we deploy these plankton nets so that we can capture organisms and we can do what we call onboard imaging. So we have a system that's just like the one that we deploy off the back of a boat. So it has a camera on one end, light source on the other end, but we're able to put things in jars in between here and take images of them. So here's just a little GIF of a Deliolid that we took from that onboard imaging. And we get really cool images from these as well. So I just wanted to show a few more examples. This one here is a ctenophore, so you can actually see all of those little teens moving down the sides of its body. The little copepod jumping in there towards the end. This is a ring of salps. So again, salps, other types of pelagic tunicates, and they form chains or rings sometimes. And this is one of my favorite gifts that we got. This little guy, this weird looking alien thing, is actually a larval lobster. So if you imagine what a lobster looks like, this is what a baby lobster looks like. And he is bringing a little jelly with him for a ride, whether he's just hanging out with it, whether he's going to eat it, unclear, but he's hanging out with that jelly. And so moving forward, like I said, for this Deliolid project, we go out monthly. I'm still working with a lot of the data from the Gulf of Mexico, trying to determine where those different groups are and why they're there. But I'm going to keep asking the same questions. Who is out there? Where are they? Why are they there? And then bigger questions, how are these things potentially going to change with climate change? And not only what impacts do the environment have on the organisms, what impacts do the organisms have on their environment? And I just wanted to quickly thank some of my funding and my current lab, which is the Zero C Lab. The Deliolid project that I'm working with right now is NSF funded, and the temperature experiments that I talked about were funded through my PhD at UNC Wilmington. And here's a bunch of my contact information. If you're really into Deliolids, which I hope everyone is at this point, we have a really cool blog. It's this website right here, and I can probably give more of these links to you guys if you are interested in them. And I have a Twitter and an Instagram where I post a lot too, especially when we're out at sea, and a lot of these cool images if you're interested. And with that, I think both Shannon and I will take questions.
Shannon, do you want to join us back up here? Thank you so much to our speakers, Shannon and Laura. We do want to open it up to questions both in person and virtually. So if you have any questions, let me know. Raise your hand, I'll come on over. And we, I think, do have a few virtual questions as well. We'll start here.
So my question was that if jellyfish or jellies are made out of 95% water, what makes them a good food source?
So if we're talking sea turtles, let's just use that as an example. So the leatherback sea turtle in that photo that I had, they almost the entire diet is jelly. So yeah, that doesn't seem very nutritious. But a lot of these jellies will have other organisms growing on them. So the leatherbacks love cannonball jellies, and cannonball jellies are known to have spider crabs that live on them when they're in the younger phase, and then they go benthic. So they go to the ocean floor afterward. So I would say those other organisms, along with the jellies, helps give them a better nutritional profile.
Is there a way to track or follow a particular jellyfish?
Like an individual? I would say probably not. I have heard of people trying to tag some of the more robust species before, but I don't know how successful that would necessarily be. And most of their lifespans are pretty short as well, so I don't know how much information you would actually get if you were able. Yeah, it depends on the species. I mostly just worked with polyps and Ephira and then sacrificed them when I was finished. But Shannon raises them up so she can probably.
I would say definitely in captivity a year on average. I've gotten them to live 2 years before, but they start they start to shrink and they look pretty shriveled at a certain age. So 1 year is a good round number for their age.
We do have a question from our online viewers. Jock asked, are there jellies in the open ocean away from the coastline?
Yeah, that's a really great question. There are jellies pretty much everywhere in the ocean. There are actually some species that don't have the polyp stage. So there is a species called Pelagia, it's a genus, that they have their whole life cycle in the open ocean. And so they don't actually need that benthic polyp stage that's going to be on some sort of hard substrate to go through their whole life cycle process. Can you think of any other ones? Yeah, they're pretty much everywhere.
So Bethany asked from our virtual viewers, have you seen any trends with fish and jellyfish relationships in your work? Laura, I think this is for you.
I haven't specifically because I haven't been looking for them. It's mostly just anecdotal where I'm sorting images and you see a lot of images that have the fish associated with them. It's definitely something that I thought about looking into, but there just hasn't been enough. The jellies, especially in the Gulf of Mexico, where I'm working with a lot of that image data, they're pretty sparse, some of the groups. And so the ones that have fish associated with them, there's a lot of them, but they're even more sparse than the actual jellies. So I would have to really dive into that more, but I've definitely thought about it.
I can just tag on to that. So just collecting offshore, we see a lot of fish interacting with the jellies like I showed in the photos, and it's always usually larval fishes.
Oh, thank you.
Most of most of the time, I think that when we see a jellyfish, it's in the sand, like we were saying. Is there any way, or is there anything we should do in terms of trying to get them back to the ocean, or just leave them?
So once they wash up, so I was saying they're so delicate, that does a lot of damage. That's pretty irreversible. So once they wash up, that's pretty much it.
And I would say, especially depending on the species, I wouldn't touch them, at least not with your hands, because they can still sting you once they're dead. Those stinging cells are active. I'm not sure exactly how long, but they're active for a while once they're still dead. So you can get stung by a dead jellyfish if you touch the tentacles and those stinging cells get into you.
And just to add on to that, the tentacles can rehydrate. So if you don't remove that tentacle from your skin and you get your skin wet again, you can get stung all over again. And you don't want to take sand and rub it in. That will just make it worse.
So I think a lot of the lifeguards around Charleston have a vinegar spray for jellyfish stings. That's one thing that can help with it and kind of scraping that off. We do have another question from our online viewers. Eloise asks, how do the fish that use some venomous jellies as protection not get stung?
They just stay away from those tentacles. So fish swim much faster than jellyfish do, so they instinctively know to stay away.
Cool. Do we have another question now?
Kind of tagging on to that one, when they have the symbiotic relationship with the jellies, are they also still avoiding the tentacles, or is it similar to like a clownfish and an anemone where they have mechanisms to not get stung?
So that could be. I'm not aware of it, but I do know those fish can still get stung if they come in contact.
Any other questions from our in-person audience? Wonderful.
Woo, yeah.
Certain times a year, it seems like there's huge quantities of jellyfish on the beach. Is that a temperature, weather? What's going on that causes so many to wash up?
Yeah, I would say it's probably temperature that's causing them to go through the process of strobilation. There's also a chance that just depending on the currents and what's going on with the tides at the time. You can kind of think that there's always jellies out there, just whether or not they're kind of all being forced into one location. So there might have not been a certain temperature event that caused them to actually reproduce quickly. Maybe all of those adults were already out there, but something with how the currents and tides are going kind of forced them all into one location, and then thus they wash up on the beach. So it's a little unclear. We know that temperature plays a role in their reproduction, but sometimes when it seems like those blooms come out of nowhere, it could just be that they had already reproduced and then something physical kind of aggregated them all together.
And just to add on to that too, if you have jellies in the water, just because they wash up together, that may not have been a bloom event. It could be, like she was saying, the wind, the tide, the current pushed them all together, congregated them in the one area, and then remember, they can't swim very well. Box jellies, cubozoans are the only ones that can swim a little bit stronger, and that's not even a very strong swim. So if they get too close to the shore and the winds are right, it'll just push them right on her.
Wonderful. I want to say thank you again to our speakers, Shannon and Laura. And I also want to say thank you to our sponsors, Mary and Mason Holland and Chris Cocker of Ameriprise Financial Services. And I do want to let you all know about our next Holland event. It will be on October 19th, all about bobcats with Jim Jordan from the town of Kiowa. So we hope to see you then, and I hope you have a great rest of your evening. Thank you.